Rejuvenation. Say it 1,000,000 times, then say it 1,000,000 more, and the word you will have said 2,000,000 times is... No, rejuvenation, because that's what we're talking about today. Specifically how this relates to human rejuvenation and the three main strategies. Why? Well, in case you haven't heard the news, Altos Labs has taken over Twitter, and they're going to be doing some cool work, as are many other researchers and other companies that have also entered the space. But before we dive into the three main strategies, what do we even mean by rejuvenation? Well, rejuvenation refers to reversal of aging, so it differs from strategies that target direct causes of mortality, and also from strategies that slow down or attenuate the biological age process. I've always since I first saw this figure, liked this figure here because it demonstrates how rejuvenation differs from duroprotective mechanisms. If aging is akin to gravity, then like the flow of water, duroprotective strategies are like dams and canals that slow down the flow rate, whilst rejuvenation strategies are more like water pumps. However, it may not be that clear-cut for some strategies as to whether it is duroprotective or rejuvenating. One may see signs of rejuvenation at a single-cell level or a tissue, but at the organism and functional level, such as frailty measurements, it may appear only to slow down the process. Secondly, we would need reliable ways of measuring biological age to see how these strategies impact it. But anyway, enough with semantics and definitions, let's look at what are considered the three main rejuvenating strategies. So firstly, factors in the blood or not. Some of the earliest indications that we could manipulate aging came from studies conducted in the 1900s known as heterochronic parabiosis, simply surgically joining two animals together such that they share a circulatory system. Sounds a bit grim, but it provided an experimental system to test for systemic effects on aging in different tissues, since the circulatory system is body-wide. The earliest work was published in the late 1950s and early 1960s, where here they could perform the experiments by combining young and old rats. A more systemic study conducted in 1972 showed that when older members of a female heterochronic pair outlived single female controls and homochronic female control parabionts. So this hinted that there might be something in the young blood that was enabling these older female rats to live longer. But as correctly pointed out in this study, that increase of life expectancy is not equivalent to decelerated aging. The younger member of the heterochronic pair may enhance survival of the older by protecting it against disease, not directly attributable to senescence. But much of this work has now been revisited, now with newer techniques and resources. And for a while it was thought that there must be factors in the young blood that have declined in the old. This stemmed from a 2005 publication by the Conboy Lab where heterochronic parabiosis ameliorated aging phenotypes, and in a study in 2020 showing that young blood plasma fractions reversed epigenetic age in rats. So with these tools we can now see age reversal. It may be a relief to hear that in this latter paper they didn't have to fuse the circulatory system together. And another approach that also avoids this parabiosis problem is plasma dilution. This builds on the idea that the benefits from heterochronic parabiosis may have also come from effectively diluting the bad factors in the old blood. And work again coming from the Conboy Lab showed this in 2020, whereby diluting aged plasma in mice with saline and albumin rejuvenated the three tissue lines. So they saw signatures of cell replication, which more closely matches what is seen in younger mice. There is still more data to collect though to really evaluate if this is age reversal, but since replacement of blood plasma is already something pretty safe, though I guess time-consuming in humans, it hopefully shouldn't take too long to see these results. But it is worth mentioning that it is very likely that a combination of both additional factors from young blood and dilution of old blood is beneficial. But as I said, more work will be needed to tease these two aspects apart and to further confirm the presence of a rejuvenating effect. So to the second strategy, cellular reprogramming. Now, many of you probably associate this with the Yamanaka factors, and rightly so. Back in 2006, Shinya Yamanaka published a now landmark study whereby the introduction of 4 factors, the Yamanaka factors, OSKM, when expressed in differentiated mouse cells could reverse them to pluripotent stem cells. Cells that have the potential to divide continuously but also differentiate. So they were called induced pluripotent stem cells. Hence, this is where reprogramming comes in. You've effectively altered a cell's identity. And so by measuring the age of these reprogrammed cells, again here using epigenetic age, which is based on DNA methylation marks, a decrease in age was seen. So that's pretty cool. We can turn a specialized cell into something younger, but what does that mean? That's just one cell. Well, a 2020 paper showed that the use of 3 Yamanaka factors, OSK, allowed old mice to regain eyesight, and a new preprint has shown that a single transient expression of OSKM in naturally aged mice reversed aging in the pancreas, liver, spleen, and blood. I'll cover this work in more detail when it's officially published, so you want to make sure you subscribe so you don't miss that. But moreover, you can use these induced pluripotent stem cells to make entirely new embryos. But this latter paper reinforces how unleashing this replicated potential also comes with its own risks, cancer development. And so ultimately, to get better control over these exciting mechanisms, it is best to understand how reversing the identity of a cell could reverse age. And can other factors or combination of factors be found? And will that combination vary depending on tissue type? And so instead of Yamanaka factors, you may now hear them referred to more generally as reprogramming factors. But small molecules potentially could also work. And so as you can probably tell, there are many unanswered questions, which is part of the reason why there's been so much excitement around the Altos Labs announcement and the rise of more rejuvenation companies. And so the last strategy, understanding the endogenous. Now, this strategy is a little different, but nonetheless still interesting, partly because there is much still left to uncover, and is the reason why all of us were young when we were born. That is, the natural rejuvenation that must occur after fertilization to erase damage from the egg and sperm from our parents that give rise to our fresh-faced selves. Recent development of the first single-cell epigenetic clock, single-cell age, has also provided data now to support a rejuvenation process in early development. And so we know that this is a rejuvenating event. The question is, what are the molecular mechanisms involved? Part of this age reversal may come from telomere extension, as there's some previous work showing that growing mice from embryonic stem cells that have been passaged for more times in culture, which effectively meant that they had longer telomeres, enabled the elongation of lifespan with low incidence of disease. But also, as you can see in this figure, early development, there is lots of rapid division and growth, which also means opportunities for damage to get diluted. So similar to above with the plasma dilution, but on a very different scale. But yeah, understanding the mechanism could be useful down the line therapeutically. So to quote from this recent review article, much of the biological underpinnings of rejuvenation remain enigmatic, and the current side effects induced by some of these interventions, particularly reprogramming, currently prevent their translational application. So Altos Labs and the other companies have many challenges ahead. Will they succeed? Who knows? But what I am sure of is that we will uncover some interesting biology along the way. So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.